All right, so I'm going to try and go as quickly as possible here, and I'm going to get into the details of each of these chapters, unlike the previous video lectures that you've had. Those video lectures were trying to give you an idea of what was coming ahead, whereas here I'm going to be trying to give a regular lecture like I would in class. And so I will focus more on the details, but at the same time, because I am having to record so much time, it won't necessarily be as precise in terms of the language that I've used before. I might make mistakes. I might stop myself. I might say cancel that out and just keep going because I don't really have that much time to edit all of these videos for you. So chapter 26 is all about how the early Earth was and how do we think that life first formed. So there are a few key points in terms of Earth's history that we want to take a look at. Major events, major things that were happening. 12 billion years ago, solar system forms. The Earth forms about 4.5 billion years ago. We think it might be older than that, but that's the oldest rocks that we can find. Then we've got life forming some point between 3.5 and 4 billion years ago. The reason why there's no certain date is that we have a lot of chemical trace signatures or trace fossils that tell us that life was probably around 4 billion years ago that it evolved or that it formed. But the earliest evidence that we actually have in terms of actual fossils of those living things is about 3.5 billion years old. So prokaryotes are pretty much the only thing that exists for about 1.5 billion years. The first fossils that we have of prokaryotes are called stromatolites. They're basically rocks that stick out from the bottom of shallow ocean floors in sort of the intertidal zones where the water's coming in and out. Bacterial colonies form, and each year they grow larger and larger. And we have fossils of those types of rocks. So that's our first evidence of prokaryotic life, is these bacterial colonies. Oxygen is not present in the atmosphere until about 2.7 billion years ago. That's when the first photosynthetic bacteria actually start to release oxygen. In the beginning, they don't necessarily release oxygen straight into the atmosphere. There was a lot of iron in the water at that time. So iron and oxygen, when they come together, it forms rust, and that rust precipitated out into the bottom of the ocean floor. And we still have record of that. There's still a really large iron band that pretty much exists in the fossil record over all the places that were underwater when this process first started to be undergone. Keep in mind here that the bacteria that are going through photosynthesis don't have chloroplasts. They are not plants. They are not algae. They are not protists. And so they're doing photosynthesis, but in an uncompartmentalized sort of fashion. So oxygen eventually starts being released into the air, and because oxygen is reactive, it actually causes the first major extinction. Most life forms at that time would have died in the presence of oxygen, and they had to really develop ways to avoid dying under those situations. So at that point in time, some bacteria would have had natural variations where they would have had certain genes that would have allowed them to survive in an oxygen-rich environment, and by surviving those environments, they were able to produce offspring and pass their genes on to their offspring. So over a relatively short period of time, we have bacteria that are able to withstand those environments. We still see remnants today of bacteria that are unable to survive in oxygen-rich environments. We call them anaerobic bacteria. A good example of that is tetanus, which lives underneath the rust on things like nails. And so the rust protects it from that oxygen. They live underneath. You step on the nail, all of a sudden they're in your body, and that's when you have to go to the hospital and get your tetanus vaccination because you've got this bacteria living inside of you. But that's just an example of anaerobic bacteria that exists today, and they are remnants of this period of time when the Earth had no oxygen. Eukaryotic life starts to form about 2.1 billion years ago, and until about 1 billion years later, they pretty much just lived independently of other cells. But around 1.2 billion years ago, you start to have multicellular eukaryotes, And the idea is that those single-celled eukaryotes started to come together and form colonies, and these colonies eventually began to form multicellular life. Animals start to come onto the scene about 500 million years ago. There's a huge explosion of animal diversity, and most of the taxa that we are familiar with seeing, they start to evolve at that point in time. So land colonization begins about 500 million years ago with plants, and it's not until later on that we actually see amphibians moving onto land. Here's just a closer look at the same chart, highlighting some of those major points. One of the things that you can see is that in terms of the history of the Earth, the time scale shows that really humans have only been around for a very short period of time. So there's always a question of, we know that all life is related to each other because, for example, we use the same DNA code to produce our proteins. Not only that, but we use the same amino acids. There's more than just the 20 amino acids that we use to make proteins that exist in the world. But the fact that all these living things use the same amino acids to build their proteins tells us that we are all related. So life evolves once, and it never evolves again. And that sort of leads us to the question, well, if life evolved once, how did it happen? And basically, there's a 4-stage process that has to occur in order for you to have a living thing. A good way to think about this is, what would a life form, an alien life form, need to survive if it didn't look like ours? And, the basic requirements of life is that, one, you can pass down your genetic information from one generation to the next. And the second thing is that you have a compartment whereby only where the internal environment is very different from the external environment. And so we're used to seeing that with, for example, a cell membrane. So those parts are important in what makes something living, is the ability to internalize an environment and have the conditions inside different from the outside, and then also being able to pass on those genes to their offspring. So there is other stages involved in that process, though, like how do you go to the point where you get something that's actually living? And that's sort of where we have these 4 stages coming into play. So the first thing that has to happen for you to have life is the abiotic synthesis of organic compounds. What does that mean? It means that you have to have a non-living process to create the things that will form the very foundation of life. So in the early Earth's history, there was no glucose just floating around in the water. There was no, phospholipids just sitting there in the surface of the rocks because of living things having left them there. They had to be created through some chemical reactions that would be spontaneously occurring in the environment that originally existed in the Earth. The second thing that you need to do is take those monomers, take that glucose, and turn it into something that's a polymer. So turn the glucose into, for example, cellulose, or turn the glucose into starch. There has to be a way of taking amino acids and turning them into proteins. And all of this, again, is happening without actual living things participating in these reactions. Once you start to have polymers, then what you need to do is have molecules that can replicate themselves. So this is important because we know in our cells, our DNA needs to replicate itself to be able to produce the instructions necessary for the next generation of cells. Now, in this case, that has to happen independently of being inside of the cells before we can actually have it happen inside of a cell, inside of something that's living. And then the 4th step is taking all these parts and putting them together into something that's a protobiont, which means like an early life form, something that wasn't living but was kind of alive. And eventually we have these protobionts at some point acquired all the necessary conditions to form something that was living, and the ability to control the internal environment And the ability to pass on instructions from one generation to the next was so overwhelmingly advantageous in this environment that those cells that were actually alive were able to reproduce and pass on their genes to their offspring. And so we have all life descending from this one early life form that had evolved all the prerequisites for something that we would consider being alive. So there's some experiments where people basically tried to take a look at this and see if they could replicate the same conditions that had existed in early Earth. And so we have the experiment conducted by, sorry, I'm going to just skip that one and go to the Miller-Urey experiment, where basically what they tried to do was see if the conditions in the early Earth were, favorable to towards the creation of these organic monomers, so creating things like glucose, amino acids, nucleotides. So what the Miller-Urey experiment does is they put into this chamber that you can see here all the things that they thought existed in the early Earth's history. So they put water, hydrogen, methane, and ammonia into this mixture. They then heated up the mixture, simulating, for example, a volcanic reaction, creating vapors in an ocean nearby, or any sort of process that would have sent this stuff up into the atmosphere. What they then did is, as this mixture went into the early Earth's atmosphere, they then sent an electric shock through it, which was supposed to simulate lightning. And what happened was, when condensation occurred, this fluid, this mixture that they got out down here at the bottom, contained all 20 amino acids. It contained nitrogen bases and it had ATP. So using very basic chemicals that they thought existed in the beginning of early Earth's history, they were able to get all the precursors for life, all these monomers. So that's why that experiment is important. The problems with this experiment is nobody really knows what the early Earth's atmosphere consisted of, other than the fact that it didn't have oxygen. There's a lot of debate whether it had methane, whether it had ammonia, and obviously that would change the results of this experiment if some of those molecules weren't found in that early Earth mixture. The thing that we also aren't sure about is, where does that energy source for these chemical reactions come from? Does the energy source come from volcanic reactions? Does it come from lightning? Does it come from UV light? Remember that early Earth's atmosphere did not have oxygen, so without oxygen you can't create an ozone layer, which means that UV light is free to come in. Remember that ozone is made out of O3. So at that point I'm going to stop the video, because I think I'm going to be running long here on uploading it to YouTube, and I'll continue with this lecture here in a moment.